All right, looks like we are live at Science Bubble. We have a few attendees jumping in. Let's see. Are we up on Facebook, Madison and Oksana? Let's see.
I don't think so. I couldn't see the notification for it.
Okay. Oh yeah, I see. It's actually still going. Maybe I'll relaunch. Okay, and then let's test the chat to make sure the chat is working. All right, thank you for those who have joined. All right. Okay, so thank you so much. Just giving a few moments. Looks like Facebook is not live streaming. Okay. Okay. All right. Well, I'm going to go ahead and read our land acknowledgement. Thank you for everyone who's tuned in tonight. And we'll just take a moment as people are logging in. So this is the Berkeley Public Library, and this is our monthly Popping the Science Bubble program. And the Berkeley Public Library has a land acknowledgement, even though the program is a virtual program. We like to acknowledge the land that the library physically itself sits on. So the Berkeley Public Library acknowledges that we sit on the unseceded traditional lands of the Ohlone peoples. We honor with gratitude the land itself, as well as the Ohlone peoples who still live, work, and play in this region. The Berkeley Public Library condemns all forms of hatred, racism, and other forms of discrimination. We are outraged and saddened at the surge of hate crimes towards Asian American communities, as well as the racism that has been shown towards African American communities. We stand in solidarity with all marginalized people that have been subjected to hatred and intolerance, and celebrate the differences that contribute to the strength of our community. Everyone is welcome here at the Berkeley Public Library. All right. And just to introduce our Science Bubble team, Popping the Science Bubble, welcome. This is a monthly seminar series that aims to share new research findings from grad students and postdocs at UC Berkeley with the general public and create constructive discussion about a variety of science topics. We have 2 speakers at each seminar who will talk about their current research or a topic that they find really interesting. The organizers are 3 graduate students at UC Berkeley, Jenna, Madison, and Oksana. If you're interested in checking out their past seminars, you can visit their website, check out their Twitter or Facebook, and sign up for the listserv to keep in the loop for upcoming seminars. Our speakers shared additional interesting researches on WordPress sometimes, and you can learn a lot from the WordPress website. We also recommend joining the listserv to learn about upcoming topics, and going to the YouTube channel to watch this presentation or watch other presentations that the Science Bubble team has hosted. All right, thank you so much. I'll let Science Bubble introduce tonight's speakers.
Thank you, Kelsey. Hi, everyone. Thank you for being here tonight. We're excited to have you here at Popping the Science Bubble. As Kelsey said, we're a student-run seminar series bringing science from Berkeley and the general Bay Area scientific community to the public. We're here every third Tuesday of the month at 5:30 p.m. Currently virtual, of course, but hopefully soon to be hybrid in-person and virtual. So hopefully you can come meet us soon. But for today, we're still on Zoom. We're here with our speakers, Elliot and Victoria, and our format is open for questions. So if you are curious about the topic or ask for a clarification or otherwise have a question that comes up in the middle of a talk, please feel free to enter it either in the chat or the Q&A section or the comment section on Facebook if that's where you're watching. And we will monitor these formats in real time so that we can get your questions to the speakers as they're giving their talk. So without further ado, I'd like to introduce Elliot Reynolds, our first speaker for the evening from the Lawrence Berkeley National Labs and CERN. Elliot grew up in Shrewsbury, which is a medieval market town in the UK, and got his undergraduate degree from the University of Oxford and then his PhD from the University of Birmingham. He spent 2 years working as a postdoctoral researcher at the University of Birmingham and is now working as a Chamberlain Fellow at the Lawrence Berkeley National Laboratory for about the past year. And outside of research, he enjoys sports, music, and traveling. We're excited that he's here in Berkeley with us to give this talk. So I'll stop sharing this screen now, and Elliot, you are free to share yours and take it away.
Brilliant. Can you see the slides and hear me okay?
Yeah.
Marvelous. Okay. So the talk will be fairly generic, covering particle physics, a lot of the motivation behind what we do. And then I'll end by talking briefly about my previous and current research. Yeah, the talk is called Particle Physics, the Higgs Boson and the Theory of Everything. And let's jump straight in with the theory of everything. So in order to get an idea of a theory of everything, a theory that is supposed to describe the entire universe, a first stepping stone might be to ask what the world is made of. And to do this, let's take a simplified description of a human being. So a human being is by and large made of organs, blood, bones, etc. These are made of cells. Cells are made of molecules. Molecules are made of atoms. Atoms are made of electrons and a nucleus. And now for the first time in this breakdown, one of the words is highlighted in blue. I don't know if anyone in the chat wants to try and guess why that might be the case. I'll give it a few seconds in case.
Elliot, could you possibly repeat the question?
So the question is, can anyone guess why the electrons are in blue? What's special about the electron of all the objects that I've mentioned so far? What might make it different from every other object I've described or mentioned? Ah, perfect. Okay, we have a correct answer. It can't be broken down. That's exactly the correct answer. The electron is a fundamental particle. So the electron is, to the best of our knowledge, completely fundamental. It's not made of anything else. It's not a structure like all of the other objects mentioned so far. So moving on, we can zoom in a bit further and look at the nucleus, which is made of protons and neutrons. Protons and neutrons, again, in this simplified picture, we can say are made of up quarks and down quarks. And to the best of our knowledge, up quarks and down quarks are also fundamental. And so here is a kind of conclusion that can therefore be drawn, because it's not just, the heart or not just any particular cell, but in fact, every part of a human being that this logic can apply to. And therefore, humans are made pretty much entirely of electrons, up quarks and down quarks. But so is almost everything else. So are other animals like the dog shown here. So's coins, so's every element in the periodic table. In fact, almost the entire universe that we interact with on a day-to-day basis are made of just these three ingredients. And so the answer to this "what is the world made of" question actually turns out to be fairly simple, at least at this point. We're going to make things a bit more complicated again in a moment. So we've got these three particles: the up quark, the down quark, and the electron. There is one other particle that contributes to what we call the first generation fermions. You'll understand the first generation thing in a second. Fermions mean that they have this quantum number spin, which is a half, but I don't think we need to discuss that in any detail. So the final particle of the first generation is the neutrino, which was named by Enrico Fermi, and it roughly translates to the little neutral one. And it's something like a partner to the electron, but it's very neutral. That means it doesn't feel most of the forces, so it kind of just whizzes by. So something like a million neutrinos, if I recall correctly, go through your body every second and you don't notice. So they're kind of like, you can think of them as ghosts or something to all intents and purposes. They don't tend to interact with things. They just float straight through at nearly the speed of light. Now, this is not all there is. As I said, the picture gets more complicated. As well as the first generation fermions, we in fact have a second generation: the charm quark, the strange quark, the muon, and its neutrino. Now, these are all heavier than the first generation, except possibly for the neutrino, where we don't really know their masses. And then there's a third generation: the top quark, the bottom quark, and the tau lepton, which similarly are very similar to the first generation, again, but even heavier than both the first and the second generation, again, except possibly for the neutrino. However, that kind of covers what the world is made of. But if we're to have a theory of everything, it shouldn't just tell you what the world's made of. It also has to say how these things work. So if the whole world is made of particles, we've answered the question, what is it all made of? So now if we can just answer the question, well, how does it all work? We're getting close, if not to, a theory of everything. And so we tend to, we historically thought about things like forces causing a mass to accelerate, shown in this picture. But we can ask the question, well, how does that happen? How do particles make that happen? And we've made some progress on this question. So particle physics does not just describe what the world is made of, but in fact how reality functions on the most fundamental level. And it does so through things that we call interactions or forces. And the interactions we represent in some respect with these diagrams called Feynman diagrams, named for Richard Feynman. Now, these diagrams are not just nice images, but they in fact represent probabilities or parts of probabilities in some sense. That allow you to calculate the probability that certain things will happen. And so it's through diagrams like this, at least that's one way, that we're actually able to calculate how particles evolve, how a system of particles actually works, how it interacts. And by system of particles, I could mean anything from a cloud to a table to a dog. And so these particles and their interactions really do generalize to pretty much everything. And that's what I tried to capture in this bullet point here. But reality is nothing but particles and their interactions. So in principle, the reason for absolutely everything must reduce to particle physics. And this is what is meant by a theory of everything. That's what I mean when I say that. You can break down any object to particles and their interactions, and any process, any behavior, any event to particles and their interactions. So how do they actually interact? Well, they do so through some forces, and we're going to start with 3 on this slide, but we'll come to another later. So the first is the gluon. The gluon causes the strong nuclear force, which holds nuclei together, and it's responsible for most of the collisions at the Large Hadron Collider, which I'll say something about in a moment. Now, it is associated with something called an SU3 symmetry. Now, I'm not going to try and explain what that means. To all intents and purposes, it's just an abstract mathematical concept. I don't think it's really important for this purpose. But the point I want to try and get across here is that in particle physics, there is this beautiful relationship between symmetries of the universe and forces. And the forces that exist in the universe aren't completely arbitrary, but are fundamentally limited by these symmetries in some way. They're associated with symmetries, I should say. So I just want to get that across because I find that to be a really beautiful fact about our universe, that it's so fundamentally based on symmetries at this very low level. So the next force carrier is the photon. Now this causes the electromagnetic force, which is of course responsible for much of chemistry, biology, and therefore, almost everything around us. So really it's these four, the up quark, the down quark, the electron, and the photon, are really the answer to almost any question that you might ask on your day-to-day life, but there's more particles, as you can see here. And this is associated with a mathematical symmetry called the U1 symmetry. And then lastly, we've got the W and Z bosons. Now these cause the weak nuclear force, and they're the only massive gauge bosons. So the gauge bosons, that's just another word for the force carriers. The gluon and the photon are massless. They don't have any mass whatsoever. A mass is kind of like weight in a way. It's the property that causes weight under gravitation. The gauge bosons have mass. The W and Z gauge bosons have mass. And actually there is a specific reason why they are able to have mass, which we'll come to in a bit. But they cause radioactive decay, and they're associated with the SU2 symmetry. And so here, now we've covered all of the particles and all of the forces described in the standard model. So on this slide, we have everything that anything can possibly be made of as far as we're aware, at least within the current best theory of particle physics, and all of the possible forces that they can feel, all of the interactions, all of the things that they can do.
Elliot,
mm-hmm.
Sorry, may I ask a question?
Sure.
I come from a bit more of a chemistry background, so in thinking of, like understanding the fundamental elements, right? We made a periodic table, and then the hypothesis was, there should be elements that follow these certain properties that get bigger or smaller to fill in that table, so to speak. Is there a particle physics equivalent of we think this table has been filled, or just we haven't seen any other particles, so likely these are what is out there?
Yeah, really good question. Okay, so there were at times hints that so about particle physics is a fairly young field. So about 100 years ago, we really this picture was just not there. We knew about almost nothing in the way of particles. We started by just discovering a few, and then we entered a kind of a time that I think is often called the particle zoo, where we discovered dozens, if not hundreds, of new particles, which we fit into these kind of different symmetry groups. And we used group theory to describe these particles. And it turned out that these 6 quarks made up all of those particles, and the properties of these 6 quarks predicted what those particles would be. And so for the higher level composite particles that are made of these, there was definitely something like that. For the fundamental particles, that wasn't so much the case, except that there were at times indications that there should be some heavier particle from other things that we studied that was then discovered directly. So there's not such, I think, a deductive framework here. There was for some specific particles, but not in general. And we don't know that there aren't other particles. So we've not got this kind of empty table that we've now filled. We could definitely discover more particles. And in fact, I'll talk a little bit about that later. Does that roughly answer the question?
Yes, that was great. Thank you so much.
Marvelous. Okay, so one such particle actually that was predicted by such a mechanism is the Higgs boson. So the W and Z bosons, as I mentioned, are massive, but the gauge bosons actually can't, in the standard model, you can't just give a gauge boson mass for complicated reasons. There are problems with doing that. But the Higgs boson is able to give them mass. Also, there is a process in which two W bosons can kind of bounce off each other. And without the Higgs boson, the probability of that happening goes to infinity and all the calculations break. So for both of those reasons, we had a good idea that there should be something like the Higgs boson. And we even had an idea of where roughly its mass should be. So this, to come back to the question, is an example of such a particle where we had a pretty good idea that it should be there. And in fact, if it wasn't there, that would have been much scarier than if it was there, because it would have meant the theory needed to be fundamentally rewritten in some way. Now, the Higgs boson is very interesting, but I'm going to come back to that later because it's quite closely tied to what I do. Now, the things I've been showing you so far are pictures. They're nice illustrations, but all of these pictures represent complex mathematics, which, for the sake of brevity and keeping things, relatively easy to follow, I'm brushing under the rug. Now that diagram that I showed you with all of those forces represents this thing here, which is called the Lagrangian. The Lagrangian contains all of the laws of physics in the standard model, that is, all the particles and all of the forces. So this might look like a big, ugly equation, but considering everything that we're aware of in the universe, with some very few exceptions, is described by this equation, it's pretty short. And CERN famously does a mug that has this equation on. So if the theory that describes almost the entire universe can fit on a mug or a t-shirt, I think it's relatively simple. We're doing it's doing a good job. So particle physicists try to discover the Lagrangian of our universe. That's basically our job. That's what we try to do. And that's equivalent to discovering a new particle, often at least. So now a question for the audience again, but what's missing in this picture? We've got all of the matter particles that make everything up. We've got the strong and weak nuclear forces, the electromagnetic force, and the Higgs boson. Can anyone suggest something? Can anyone think of anything that might be missing in this picture? Give another few seconds in case someone's typing. Okay, so one answer is energy. Energy is included in this picture. Energy is kind of a complicated concept. It exists in many different forms. It exists, for instance, in the form of kinetic energy, which is just motion. That's when one of these particles moves. It exists in the form of mass, which, for instance, is just when a massive particle like the Higgs boson or the Z boson is just sitting there. So energy is encapsulated in this picture in some way, but the second answer, gravity, is spot on, exactly what I was hoping would be said. So gravity is the answer. At the moment, we use a theory that Einstein came up with called general relativity to describe gravity, but it's fundamentally not quantum mechanical. And so for this reason, we know that either the standard model of particle physics that I've just been talking about, or general relativity, our theory of gravity, has to be amended in some way for these two to be married together. And one possible way that this could happen is perhaps a graviton. That's one idea as to how gravity could be represented in a quantum mechanical way. What else? So I'll give people another, there are other answers. We've not quite got all the answers yet. The next one's probably harder though. So I'll give 5 seconds or so, maybe 10 for the answer. This one's a bit trickier. Okay, dark energy. Okay. So can we see the chart again? Okay, so here's the chart. Someone gave the answer dark energy. I was going to do this 3 times. You've got this one and this one. Dark energy is the third one. The second one I'll just give you so we can move on, was dark matter. I was assuming you get dark matter before dark energy, but I'm happy with you skipping one. Okay, so yeah, what else? Dark matter. They're both on this slide. So about 5% of the universe is made of ordinary matter. That's us, our shirt, table, basically everything we interact with, the sun, the earth. But about 5 times as much matter, as measured in the amount of energy, is made of this mysterious stuff called dark matter that we really don't know what it is. And now we have some idea as to what it might be. And we think it might be weakly interactive massive particles, or WIMPs. So when I say weakly interacting, what I mean is the same thing I meant with the neutrino. It doesn't do stuff. So if it goes through you, it just goes through you. It doesn't stop, it doesn't touch you. It's like a ghost. And massive, I just mean heavy, I mean has a lot of mass, which is something like being heavy at least. Now, one piece of evidence that dark matter is in fact these kind of very heavy, almost ghost particles, weakly interactive massive particles, is from this event that we call the bullet cluster. So these are where 2 celestial bodies collided, and the visible matter is shown by this heat map. That's mostly stars and stuff. And that kind of hit each other, slowed down in the process as it all smashed into each other. And so it went through itself really slowly and only got to here. The green lines show the gravitational matter. They show where the gravity is coming from. And you see that sped through faster. So the idea here is that most of the gravity is being caused by these WIMPs, which went straight through itself, because as I said, it's like a ghost, and ended up here and here. And the visible matter got slowed down as it smashed into each other, as would happen if, anything smashes into itself. So this is one piece of evidence that dark matter is in fact made of these WIMPs, but there are other ideas as well. And then anything else, and of course we've got the answer, that's dark energy, the other roughly 70% of the universe. Dark energy, we've got much less idea what it is. We really, there's some theories, but at this point it's little more than speculation. We really don't have a good idea as to what this is, which, considering it's about 70% of the universe, means that we do have some work to do. So other questions and problems. In the standard model, there are 26 free parameters. Now, when I say free parameters, I just mean numbers that we don't know why they take the value they are. Things like the mass of the electron. We don't know why it has a mass that it has. We don't know why it's not twice as heavy or twice as light. And that's pretty unsatisfying. Like we would hope that the final theory of everything should be beautiful and elegant and really just everything fall from one simple assumption or something like that. The idea that there's 26 numbers that we just have to measure is kind of ugly. We also don't know why there's 3 generations of particles and why they get heavier. Why not 2? Why not 4? Why not 1? 1 would perhaps seem the most elegant. Why not 7? Why not 12? We really don't know. Why not infinity? Another question is, where did all the antimatter go? We think at the beginning of the universe, so every particle has this antiparticle, which is kind of a similar but opposite form of itself. So if the particle, like an electron, has a negative charge, the antiparticle has a positive charge, but they have the same mass. And now these antiparticles, we think they were produced in the same amount as the particles at the beginning of the universe, but now the universe seems to be made mostly of normal matter, not antimatter. And we don't know exactly where all this antimatter has gone. So this is one other question that we're trying to solve. Now, the way that we try to solve this, the way we do this, is using the CERN Large Hadron Collider. That's one way we're investigating these questions, at least. Now, the LHC is the biggest machine ever built. It's built under the ground across the French-Swiss border, just near beautiful Geneva, shown here. Now, it was built and operated by CERN, which is a European nuclear research laboratory, which is currently led by Dr. Fabiola Gianotti, who's the current Director General of CERN. Now, of course, we have a big collider, and on this collider, we have 4 main experiments. Now, I don't have time to talk about all 3, but I'm just going to very briefly mention the one I work on, which is the ATLAS experiment, which uses the ATLAS detector. Now, I'm not going to say much about the detector because it would take a long time to go into detail about exactly how this thing works. But I'm going to point out the scale of the machine these two humans here are roughly to scale. So you can get an idea that this is an absolutely enormous machine. It's something around 100 meters under the ground. So this really is a massive project. And there's something around 3,000 authors currently signed to the Atlas collaboration. So it's a huge collaboration as well, and far more people working on it. But this detector, so the particles collide, the LHC collides protons at very high energies. They collide at the center of the detector. And then in these collisions, the outgoing particles are measured by the detector. And we kind of try to piece together what happened at the collision by measuring those outgoing particles and work out whether something new or interesting happened there. Now, one new and interesting thing that happens occasionally is that we produce a Higgs boson. Now, the Higgs boson is a new and unique fundamental particle, and it is also associated with its own interaction. So it's not just a new particle, as I showed you before, but it also allows particles to interact in new ways. It caused something called electroweak symmetry breaking. Now, this is basically where one of the fundamental symmetries of the universe was broken. And when this happened, the electroweak force, which was some kind of merger of the electromagnetic force that we know today, what happens when you hold a magnet somewhere, and the weak nuclear force, the thing that causes particles decay, these were actually one thing called the electroweak force. And the breaking of this symmetry that was caused by or associated with the Higgs boson broke the electroweak force into the electromagnetic and weak nuclear forces that we know today. In doing so, it caused mass to exist. And the Higgs boson is also interesting because it's the only particle associated with a field with a non-zero vacuum expectation value. Now, I'm not going to try to explain in full what this means, but basically to say that particles are associated with fields. So the photon, for example, is associated with the electromagnetic field. And when there's no photons and no magnets, nothing generating an electromagnetic field, the electromagnetic field is zero everywhere. And it only appears when you hold a magnet there or you shine a light. The Higgs boson is different. Even when there's no Higgs bosons, the Higgs field is always there, always turned on, so to speak, with a value other than zero. And it's the only particle like this, which is really very odd, very different, at least, from the other particles. It's also the only spin zero particle. So particles have this thing called spin, and it's the only particle where that spin is zero. And there are very interesting consequences of this, which I'd be happy to discuss after, but so that I stick roughly to time, that's going to have to go under the rug for now. And it also completes the standard model. So it completes all of the particles that have been so far predicted by particle physicists. So this is actually the last particle that we have a really solid prediction for, to get back to the earlier question. So now I'll just, given that context, just say a very brief thing about what I have been doing and what I am doing now. So the first thing I'll mention is that I do searches for decays of the Higgs boson to new particles, to beyond the standard model particles. So as I mentioned, there could be other particles. And at the LHC, one of the things we do is we look for them. We look for hypothetical particles and hope to find something. Two of the searches I did are represented by these two diagrams here, which were published in the searches published in these two papers. The first was a decay of the Higgs boson to two new particles, which each decay to a pair of muons, which are like a heavier partner of an electron. Then we try and detect these muons in the detector, and by using the properties of these muons, try to work out that this decay has happened and therefore that these new particles exist. In this diagram, the Higgs decays through some unspecified process to a Z boson and a new particle. The Z boson decays to electrons, or the heavier cousin, and the new particle decays to either quarks or gluons, which kind of form a spray of particles called hadrons that we detect in the detector. And there are lots of these jets of hadrons in the detector. And so we use machine learning methods to try to discern whether they came from a new particle or not. Now, I should say at this point that Atlas is a massive collaboration, and all of the authors of the papers have in some way contributed to the overall effort. And in fact, the specific analysis done here is also part of a bigger team. So of course, I'm very clear here that I'm not claiming credit 100% for all of this work. I was part of a team with all of this. So the next thing I do is that I've been part of teams trying to probe the strength of the interaction between the Higgs boson and the charm quark. And we've done this previously in the decay of the Higgs boson, where you can try to probe the interaction strength by searching for Higgs bosons decaying to charm quarks. And this was published in this paper here. And more recently, I've been trying to investigate whether you can also do this by looking for the associated production of Higgs boson and charm quarks. That's where they're produced together in LHC collisions. And this is not yet published. This diagram comes from someone else's paper. And so whether this ends up published or not is yet to be seen. Another thing that I'm involved in is probing the interaction strength between the Higgs boson and itself, which occurs through diagrams like the one shown here. And this is publicly accessible now. You can see this in this note, which will hopefully be published in a journal soon. I also have done some work working towards the upgrade of the ATLAS detector. So the LHC is getting the Large Hadron Collider is getting an upgrade, which will allow it to take data faster. But in the process, it will irradiate the detector more. And also, every time protons collide, the events will be much busier. There'll be more going on. And so we need a better detector, both to survive the radiation and also to understand what's going on when there's kind of more going on there. So more stuff overlapping with itself, with each other. And so parts of the detector need to be completely replaced. And I've been doing some work towards that as well. I also am involved in the development of some machine learning methods, both directly for particle physics for a specific search, and also more broadly, hoping that they'll be adopted for other searches. The papers resulting from this, or documents resulting from this, are shown here, and there's a schematic of one of the methods that I helped develop here, but it's quite complicated. So again, maybe we'll come back to it for the questions if people are interested. I'm probably already over time, so to quickly summarize, the universe is nothing other than particles and their interactions. So a theory that describes those is truly a theory of everything. Now, for me, at its core, particle physics is the search for this theory, for the theory of everything. The standard model is our current best model of particle physics, but it's not complete. We know that there are questions and problems that it doesn't resolve. And the Higgs boson is a new and unique particle that might be the key to answering many of these questions and ultimately to finding physics beyond the standard model. If you want to know more, there's a variety of links here with various resources, which I can provide, but I'll flick through for now and just say thank you for listening, and happy to answer any questions.
Madison, you are muted.
Yeah. It looks like we have a
yes.
question in the Q&A. So how common are Higgs particles and how often do they appear in your detector studies? Okay,
so the way we tend to think about the production of particles is through their cross-section. And the cross-section for the production of the Higgs boson is about 50 picobars. Now, I haven't done the calculation myself, at least not recently, but I think I've heard that this results in about 1 Higgs boson per second. Now, the caveat to bear in mind here is that the LHC collides protons very fast. And the amount of data produced in these collisions is far more than we can actually read out and store. So most of the collisions get thrown away. I've heard that the amount of data produced in these collisions, if it were to all be read out, is of a higher bandwidth than what goes through the entire internet in any given time period, just to give you some idea of the amount of data we're talking about here. So it's completely infeasible to read out all of these. So in practice, we use things called triggers, which are specialized electronics and software designed to very quickly decide which events to read out. And so in practice, we won't be reading out all of these Higgs bosons, so we won't actually record one every second if that's how often they're produced.
I think in the interest of time, we're just going to take one more live question, but you're welcome to continue typing out your questions. And I think Elliot will be able to reply in writing. So what is energy?
Energy is, it's a kind of slippery concept in a way, because it's not really one thing. Kinetic energy is motion. Mass energy is mass. Gravitational energy is just things being held far apart. Really what energy is, I would say, is an observation of a conservation law. That is to say that we observed that a certain mathematical quantity is conserved. The gravitational energy plus the potential energy plus the mass energy, all of these things are completely different. You know, having some kind of massive stuff is obviously very different to two things with mass being held far apart. Those are completely different concepts. To describe those as the same thing is almost nonsensical in a way. Really, the only reason that we use this one word energy and connect all of these things is that we've noticed that if we express the energy of these things, some expression, some function of the distance things are held apart, or the mass of the thing, or how fast it's moving and what its mass is. If we express that in a particular way, in a particular mathematical form, we've observed that the total is conserved. So it's not so much a thing as it is a conservation law, an observation that something never changes. Maybe not quite an easy answer, but I think that's probably the best I can do.
Great. Thank you so much. And thank you for the great talk. It looks like we already have another question, if you could answer it in the chat. And now we're going to move on to our second speaker today, who is Victoria. She is the 5th-year PhD in the Portnoy lab at Cal. She studies infectious diseases and focuses on the mechanism of dissemination of bacteria from the gut to the brain of their hosts. She received her undergraduate degree in biology from the George Washington University in DC, but is originally from France and moved to America for college. In her free time, she is an avid hiker and skier, and she also practices karate. We are very excited for your talk. Take it away, Victoria.
Thank you, Oksana. And let me share my screen. Can you see? Start. Can you see the slides okay?
Yeah,
Can you hear me fine?
Yep.
Okay, cool. Well, thank you, Oksana, for the introduction. And Elliot, it was really interesting. I'm actually from like right outside Geneva, where Large Hadron Collider is. Like I was looking and my town is like right outside the map that you showed. So that was kind of funny. But yeah, so my name is Victoria Chevée, and I am in the Portnoy lab at UC Berkeley, where we study Listeria Monocytogenes, say foodborne pathogen. And my work is really focused on how Listeria traffics from the intestines of its hosts towards their brain. And so my talk is going to be covering 3 main areas. First, we'll talk about Listeria as a disease-causing agent. And then we'll talk about the tool that I'm using in my work the most. So it's this barcoded bacteria system. And then finally, I'll give you some of my sort of most up-to-date data and what I think is happening in the model. And so first, I just want to make sure that everybody in the audience is very aware that bacteria are absolutely everywhere. I read while preparing for this that in the average American diet, we eat like 1 million bacteria a day. And when bacteria are eaten along with your food, they actually join the like millions of other bacteria that are already within your gut and that live there very peacefully and happily. And these bacteria are very beneficial to you. They can break down food and toxic items that you're not able to do yourself. They can make vitamins that you also are not able to produce yourself. They can help train your immune system, which are the cells that are involved in your defense mechanism against diseases. And they also just take up space so that when harmful bacteria show up in your intestines, they just don't really have everything they need to be able to thrive and grow locally. However, sometimes there's some dysregulation in things. And so harmful bacteria are still able to take a hold within your intestines. And then if they're able to cross outside of your intestinal barrier and spread to other parts of your body, that can be really problematic. And so one bacterium that is able to do that is the one I study, Listeria Monocytogenes. And it can be found in a variety of common foods, such as unpasteurized milk and cheeses, in fruits and vegetables that are raw or poorly washed, and then in fish and meats that are sort of raw or uncooked or undercooked. And so these are things that you might actually eat on a really frequent basis. So you might have eaten Listeria at lunch today and you will eat it for dinner tonight. But as somebody who has a very strong and healthy immune system, don't really realize it even happens. It just passes through you. In certain cases, it can lead to intestinal disease. And so at that point, we're talking sort of mild vomiting and diarrhea, but it passes in like 2 to 3 days. In certain cases, however, it can lead to really serious disease. And so in pregnant women, Listeria can cause miscarriages. And then in individuals who are over 65 or who are immunocompromised, so who have sort of a weakened immune system, there's a chance that Listeria can spread to a variety of organs, including the brain, and cause permanent brain damage and be lethal. And so there's about 1,600 cases of Listeria every year, and 260 of these can be lethal. And so it's the kind of disease that it's around us all the time, and it's generally really okay. So you don't have to worry about it. But if you get really sick, you should go to the hospital, and it is something to really be careful about. And so most of my work focuses on this part of the infection of Listeria, how it gets into the brain. And so the main question I'm trying to answer is, how does Listeria get into the brain? And this is the schematic I'm going to use to tell you about what we already know. So this sort of yellow, can you see my pointer? Thank you. This sort of yellow blob, that's what I'm representing as the brain. And then the brainstem and the spinal cord, there are two other parts of what we call the central nervous system. And so this is where all of your neurons are and where all of the processing of information happens. And then in blue and green, we have basically nerves that are connecting your central nervous system to all of the rest of your body, because all of your body is innervated and has connections to your brain. We have the intestines that we've discussed already. The bone marrow is the space within your bones that actually harbors immune cells that are developing. So there's the cells that are responsible for defending you against diseases. And then in red, we have blood vessels, which make this network that your blood and your immune cells can use to go just about anywhere inside your body. And so I want you to think of the brain as this sort of castle that Listeria is trying to get into. And there's 3 ways that we know it's able to get in. The first one is described as the Trojan horse transport. And so the idea is Listeria is getting into your system and it can cross the intestinal barrier, right? And then it spreads to other organs, including the bone marrow. And so in your bone marrow, there's these cells that are developing, and Listeria is able to invade those cells. And then because those cells are immune cells, they're your defense cells, and there's infection happening in all of the rest of your body, they're being called out into the circulation, into your bloodstream, to go places into their defense mechanism role. But when they're doing that, they're actually taking Listeria along. So Listeria is basically hitching a ride with those immune cells as they're going other places in your body. And then some of them will eventually find their way to be right at the interface between the blood and the brain. And Listeria has this really cool ability to do what we call actin-based motility. So it can hijack part of the cell mechanisms and then make these rocket tails. And it can basically zoom around within the cell, but also between cells. And so Listeria can use this method to zoom directly from these immune cells into your brain. Another option is direct entry. So you can think of this as basically scaling the walls of the castle. So you have free living Listeria in the blood, and they're able to use the zooming mechanism to get directly into the brain from that blood-brain interface. And then the third option is intraneural trafficking. So you can think of this as finding a side door entrance into the castle. And so we said earlier, Listeria is getting into your intestines, and then it spreads other organs. But all of your organs are innervated. And so there's nerve endings everywhere that Listeria can just invade. And then it can use your nerve and spreading from one cell to another, as basically a highway to get directly into your brainstem and spinal cord. And then from there, it can make its way into the brain. So this is what we know. And when I'm doing these experiments, I can see the brain and there's bacteria there. But one thing that's complicated for me is that when I see them, it's basically just a snapshot. I don't have a good idea of where they came from or what happened between the time they arrived and the time I'm looking at them. And so this is something that I'm trying to parse apart. And to do that, we use this tool called STAMP, which is barcoded bacteria that I'm going to describe to you in a second. But to sort of contextualize it and help you make sense of the tool, I'd like to use this analogy. So we're at the grocery store and we're trying to buy apples. And I know this may be a hot take because a lot of people actually at Berkeley know their apples really well. But to me, an apple is an apple. And then you show up and there's like 16 different types of apples and I really don't know what to do. So I always just buy a bunch of apples and I get to the place we have to pay and it looks like there's just a bunch of apples. But when you have to pay, every apple just costs a different amount and they're all actually different items. And so we use a scanner with which you can scan the barcode of each and every apple. And then you have a real count of how many Granny Smiths you had, how many Golden Delicious you had, how many Galas you had. And you have a real assessment of the proportion of all of the apples that you had within this entire group that you bought. Well, it's the same we do with bacteria, except this time we use DNA as our barcode instead of this little like string of bars. Just a little background on DNA. DNA is the genetic material that holds all of the information that encode, form and function, and that is passed on between generations. And it has this backbone, and then these chemical bases. So we have adenine, thymine, cytosine, and guanine, and they're generally represented as ATCG. And so our barcodes are 20 of these bases in any order. And so these little 20 base barcodes are placed in a part of the genome of Listeria which it doesn't affect anything. So we have all of the bacteria that are all the same, and they're happy, they're living their bacterial life, they're infecting you, and they're thriving. But they have this little sequence in their genome that allows us to identify them. And so when we start an experiment, we have something that looks like this, which is what we call a library. Each little oval represents a bacterium. And so all of these blue bacteria are going to be clones of each other. So they're all Listeria, they're all normal wild-type bacteria, but they all have this blue barcode. And then all these green ones, they're the same as all the other ones, except they have this green barcode, right, which is just a series of 20 bases. And so this way, we're calling these blue ones clones of each other, and the green ones are clones of each other, right? So we have actually 200 clones within our library, and in our library, we have a perfect representation of all of these clones. And so then when we give this to an animal, what comes out is something that looks like this. It's just a jumble of bacteria, and we have sort of no idea what it is when we're looking at it, which is what I was mentioning earlier. So what we can do is use a sequencer, basically the same as a scanner. It's going to look every bacterium, check for that barcode, and assess which one it is. And so what it gives to us is basically a readout of how many blue bacteria are there, how many purple bacteria are there, how many pinks are there. And with this information, we can make inferences about the trafficking of Listeria through different parts of your body. And so using this method, people have discovered really interesting things. And so I want to give you this example to sort of put us in the right mindset. So they gave the library, so this perfect combination of a one-to-one proportion of all the 200 clones to mice. And then after a certain amount of time, they took their liver and spleen. And so these are organs that are in your abdomen, in your belly. What they were able to find is that almost all of the clones from the library were present in these organs, suggesting it's really easy for Listeria to get there. But then when they looked at the gallbladder, which is another organ that sits just above your liver, they found that it only has one clone. So it's really hard for Listeria to get into this organ. We call it has a tight bottleneck. And then when they looked at the feces over time, the feces actually start really similar to the library. So it's really diverse, really polyclonal. And then as time passes, it becomes more monoclonal. So it only has one clone. It's really, really dominated just by this one barcode, basically. And they found that this barcode is the same one as what's in the gallbladder. And by looking at this, they were able to say basically the gallbladder acts as a reservoir, so as a hub where bacteria are able to grow. This one clone is able to do really well, and then it spills over into your GI tract, and that leads to the dominance that we see in the feces. And so what we're hoping to do is now looking at the brain, trying to understand, is it easy to get into the brain? Is it actually difficult? Is there a tight bottleneck? Is there similarities between different organs? And is there a change over time within the population? And so we started doing this. And so one of the ways we're going to be looking at the data is here on the left. So there's something that's a little bit off. The right stuff wasn't supposed to show just yet. Hopefully it doesn't mess up later. But so the way we're looking at this data is on the left here. So we'll have the organ or the sample.
We have a question.
Yeah.
So
already.
what is different between these clones? Or is it one original clone overtakes the rest scenario?
And here?
I think so, yeah.
Yes. Yeah. So that's a really, really great question, actually. I get it all the time. And so, yes, they should be exactly the same. Right. So the only thing that's different about all of these bacteria is the barcode. And the barcode doesn't affect anything. So the only reason one of them is supposed to be able to do better is basically it's a host-derived mechanism. So it's something that basically it was there's a lot of immune mechanisms that prevented other bacteria from getting there, or it got there first and then it took all the nutrients and then it was able to grow really well. So the idea is that if it is all one bacterium, that bacterium just had an advantage and then divided and led to a population, but it's probably coming just from one bacterium.
Thank you.
Yeah. So we have our sample on the y-axis, and then on the x-axis, we're going to have the frequency of barcodes. And so this is, if you look at your entire population, how often does this clone show up? Right, so here it's represented basically every clone or every barcode is represented by one color. And because it's a library, we have every single color that shows up. So we should have 200 lines and they should all be about the same size. And so now if we're looking at a real sample, so this is one mouse and we're going to have, again, frequency of barcodes at the bottom. Number of bacteria per organ is really helpful as we're trying to think about things, and it'll come up later. And then we'll be looking at the brain, which is what I'm most interested in. The brainstem and the spinal cord, which are the rest of that central nervous system, the blood, the bone marrow, and then these three organs are just a representation of what's happening in the abdomen. So they're just organs in your belly. And so what we were able to find is that the spleen, the liver, and the mesenteric lymph nodes are really diverse. There's a lot of different things happening in there. There's a lot of clones that are showing up. When we look at the bone marrow, and the blood, they're also highly diverse. But then when we look at the brain and the spinal cord, we actually see that the brain is highly dominated by one clone. And so one thing that's interesting here is that there's 94,000 bacteria in this particular organ. Right? So there's a lot of bacteria, but this one clone represents about 80% of all of these bacteria. So it's really dominant. But then in the brainstem, we have this clone that is also shared, but not as dominant. And then in the spinal cord, it's not there at all. Right? So this is what we see in a mouse at early times of infection. And then when we look later in an infection, what we're able to see is that there's a little bit less diversity in the organs and within the abdomen, right? So now there's a lot fewer barcodes that are showing up, but there's still a lot of different things. And then when we look at the brain, the brainstem and the spinal cord, they're highly monoclonal. There's only, I can tell you from the analysis, there's only one barcode in these. And so we're looking in the brainstem at 4 million bacteria, and every single one of them has the same barcode. And then when we look at the blood and the bone marrow, we find that they share that barcode with the brain and brainstem and spinal cord. But here we have a lot fewer bacteria present. So if we put all of this in a model, what we think is happening is that there's, in the beginning, bacteria are able to come into your intestines and then they spread to different organs. Here just represented as the bone marrow. And then one bacterium somehow makes it into your brain. There's inflammation, more bacteria are able to get everywhere. And then as time progresses, these bacterium that made it into your central nervous system are able to divide, but the one that was there first has an advantage because it was the first one to get there. And so it was the first one to start dividing, and it just has sort of a snowball effect, where now the more it goes, the more it gains an advantage. And then it ends up being the most dominant clone within that sample, the brain. And then these bacteria would then spread first to the brainstem and the spinal cord, leading to this completely monoclonal central nervous system that we saw in the late parts of our infection. And then we think that we saw similarities between the brain and then the blood and the bone marrow, but the brain had so many more bacteria than the blood and the bone marrow did. So we think that bacteria actually can then spill over out into the blood and the bone marrow. And so the things that we talked about today are a current hypothesis for how Listeria grows in the brain. So we think it's basically a chance event. The first one that gets there has the advantage and it can take over, and then everything after that is dependent on that first bacterium that made it in. We've talked about how Listeria is an important public health concern. And so, please stay safe and try to really wash your items before you eat them and cook your meats and vegetable well. And try to talk about it with people who might be at risk. The CDC and the FDA are great resources for learning about listeriosis. I also found this other website that I recommend. It's from Harvard, and it talks a lot about the microbiome and its beneficial effects on health. And then finally, we also talked about STAMP, which is these barcoded bacteria, and how they're a great method for studying trafficking of bacteria within hosts. And with this, I'd like to say thank you to my lab, my collaborators, the mice that make the work possible, and then all of you guys for listening, and the Popping to Science bubble organizers for inviting me. And this is my lab. At Halloween party, we were gene gnomes, and we won the costume contest. And with this, I'm happy to take any questions.
Thank you so much for the great talk. We will be taking questions now, both in the Q&A box and in the chat. And I guess I can start with one. So I was wondering, does studying the path that bacteria takes to get to the brain, does that mean that we can now come up with ways to kind of stop it from getting to the brain? Or is the main defense still like making sure the bacteria doesn't enter the body to begin with?
Yeah, that's a great question. So a lot of the work we do at Berkeley is not necessarily translational, right? So yes, it would be great if we could find a way to prevent a disease, but a lot of it is just trying to understand how it works at a very mechanistic level. And so it might be that we could be able to understand how all those dynamics are happening, and we can prevent them from happening. But mostly I think this is something that's going to give us insights into how bacteria grow, not necessarily just Listeria, but more broadly about other bacteria that can make their way into the central nervous system, and sort of the population dynamics of infections of the brain. Because Listeria you can actually cure with antibiotics. So if you show up at the hospital and you're very sick, they'll give you antibiotics, and you should be able to be cleared if you're not too far into the disease progression. Yeah.
That makes sense. Thank you.
Yeah.
And we also have a question from the audience. How is it that Listeria bacteria can survive the acidic environment of the gut?
Yeah, that's also a great question. So, yes, it's a massive barrier that the host puts up against bacteria. And so a lot of bacteria die. A lot of bacteria will die specifically in the stomach, which is so acidic. But Listeria is particularly well protected against a lot of things. So it does well in acidic conditions. It does also really well, and so it can survive decently well in your stomach, and it has a variety of sort of bacterial immune responses that allow it to deal with the stress until it gets into the gut, and then it can deal with the rest of its infection pathway. But it also is really good at dealing with the cold, which normally when people try to keep food, but it's in your fridge, you think that it's going to do really well in your fridge. Listeria grows quite well in your fridge, which is one of the reasons why it's so prevalent. And it also deals pretty well with salt. So, salt is one of the ways that people keep food. Listeria can grow in quite salty environments. So it's one of the reasons why it's one of the most prevalent foodborne pathogens.
We have another question. Do you look at the impact of probiotics too?
No, that's a very cool idea. No, so in our model, actually, we in order to get the mice infected, I didn't talk about it today, but there's a lot of my work is focusing on different routes of infection. So if you do an oral infection versus an infection where you go directly into the bloodstream, and how sort of that impacts what we understand about Listeria getting into the brain. But I spent like a year getting a model of infection of the brain after an oral infection. And to get that done, we need to give an antibiotic treatment, actually. So we give this antibiotic is called streptomycin, and our Listeria is streptomycin resistant. So we do this thing where we cause dysbiosis in the gut. And that allows, I was telling you in the beginning of my talk, your gut microbiome is really helpful at preventing, as just occupying the space and preventing bacteria from showing up. And so one thing that we do to get a more efficient infection is cause dysbiosis. So we break all of those networks within the gut, basically, and then Listeria can come in and do much better. But using probiotics is a great idea to prevent even more an oral infection with Listeria, or maybe there's something else that might somehow promote infection. I'm not sure. Yeah.
Thank you. Maybe we'll wait for one final question.
Okay.
I can also ask kind of a more maybe naive question. I was wondering at the beginning, you mentioned, having like beneficial bacteria and harmful bacteria. And I was wondering if there's, I guess, what makes bacteria good or bad? Is there like some sort of intrinsic property?
Yeah. So there's many things. There's some bacteria that are just, they will never hurt you. They're just living there and they can take advantage of the environment, the very unique environment that is the gut. And bacteria live in communities, right? So depending on who else is there, they'll do better or worse. But a lot of bacteria in your gut are sort of opportunistic pathogens. So basically they live there and they're fine. But then if something gets out of balance, suddenly they're able to take advantage of that and then they become harmful. Right. So there's some bacteria that are just harmful. Right. So Listeria is just harmful, and then it can just pass through you, but nobody has just Listeria living in their gut in their steady state. So there's sort of these three types of just good, just bad, and some that are able to sort of flip back and forth.
Got it. Thanks.
Yeah.
Okay, we have one last question. Does pasteurized milk prevent Listeria in cheese?
Yeah, it's supposed to be much better. So non-pasteurized milk, when I was looking up like all of the most common sources of foodborne infection, pasteurized, non-pasteurized milk and cheese was a really big one. And so if you pasteurize cheese, right, you apply enough heat that it should kill all the bacteria that are in there, but then you're going to keep your cheese probably in the fridge for a while. And so if at any point during the processing, there was a contact with Listeria after the pasteurization, that might be problematic. Right now, there's actually an outbreak in Florida, and it's associated with ice cream, the big Olaf ice cream in Florida. There's been 26 people who got really sick and hospitalized, and one person has died. So it is something that's still very current. Yeah.
Thank you so much. Thank you so much to both of our speakers. And if there are any lingering questions for either of them, feel free to email them to us and we'll make sure to pass them along.
All right. Thank you so much. These were both really interesting talks and both did a great job. Thank you so much. Just as a reminder, we do this program every Tuesday, third Tuesday of the month, 5:30 p.m. on Zoom. Hopefully one day in person again too. Thank you so much, Victoria and Elliot. And be sure to watch the talk on YouTube. If you want to share this video with anyone else, you can find it there. And you can also watch past Science Bubble talks as well. Have a good evening.